Hello everybody, we'll talk about the olfaction and gustation. That means the perception of smell and taste. Olfaction is supposed to be the most oldest sense, probably the first sense endowed to primordial creature. And olfaction is also considered as the most penetrating sense, the most powerful effect on emotional state, and very good olfaction is this perception is very good at triggering memories. However, the perception of smell or olfaction is not very highly developed. In many creatures, this perception is more developed than the human. So, if you just see the dogs or moths or many other creatures, they rely on the olfaction or smell a lot. And it is believed that all the creatures as well as the human used to depend on the olfaction before they develop their sight and hearing. So that's why we say that the oldest sense. So both olfaction and gustation, these are the chemical perceptions. That means the chemical molecules activate the receptors. So smell molecules activate the olfactory receptors, and taste molecules, food molecules, activate the taste receptors. So these in human, these are two chemical detection systems. Okay. In case of olfaction, the molecules are called odorants. So odorants are the molecules that bind with the receptors and activate them. Now, odorants must have few properties. So we'll talk about that. Odorants must be very small and be able to float in the air, right? So the molecules must be small. Molecules must be able to float in the air and enter into the nasal cavity, and must be water insoluble. That means lipid soluble. Okay, soluble. The term olfaction is a general term to indicate the sense of smell. Sense of smell. Okay. Now, odor, the term odor is a little bit different. Odor is a general smell sensation in a particular quality. For example, the cake had a chocolate. odor. So cake itself has a smell. However, in it we can say that this cake has chocolate odor. Okay. So the receptors for olfaction are located inside the nasal cavity. Inside the nasal cavity, you have the olfactory epithelium in the roof of the nasal cavity, and the epithelium has the olfactory receptor cells. Now, the peripheral organ for olfaction or olfactory system is the nose. So nose, a part of the nose we can see from outside, that is the external nose. However, inside the nose you have a cavity which is separated by the septum, and by using the sense of smell, the creatures, different creatures depend on the smell at different, degree, and the smell helps to detect the predators. So some creatures can, detect the predators by smelling. Different territory, recognize other individual, find food, and mates. So many creatures, they live in the forest or in the water. They depend on their olfaction for their survival and reproduction, finding food, right? Um, Identifying the others, like prey or predator. So odorant molecules, as I told you, must be lipid soluble. However, those creatures live in water. In those cases, the odorants or the smell molecules should be water soluble. Except them, odor molecules or odorants are lipid soluble, should be lipid soluble. Okay. The perception of smell or odor can varies based on the age. As we get older, the perception gets declines. Gender, usually females can detect the odor better than the males. Also, the ability to smell changes with the change of the hormones in the body. For example, pregnant women, they can smell more than the other women. Smoking can decrease the ability or can desensitize the receptors of smell. And health condition is how your nervous system is working, processing the signal. That also determines your ability to smell odor. And as I mentioned already, the odorant molecules must be able to bind with the receptors, olfactory receptors, to activate them. And when the receptors are activated, as you know, it is true for all sensory receptors, that transduction of signal will occur. And the receptors, the smell receptors are G protein mediated receptors. So G protein coupling receptors. Okay, so and the receptors are located on the cilia of the olfactory receptor cells. So olfactory receptor cells are like neurons. So neuron-like cells, this is the cell body and nucleus, and this is the axon, and this is the dendrite. So bipolar neuron, and at the end of the dendrite you have soft hair-like structures, those are called cilia, and the receptors are actually attached to the cilia. So cilia are hair-like protrusions on the dendrites of the olfactory sensory neurons or receptor cells. Okay, and the receptor molecules are attached to the cilia, and the receptors are attached to the cilia. So these are the receptors. So remember, receptor cell is a neuron. Okay, so this whole thing is a receptor cell, or olfactory receptor neuron. Okay, however, the receptors are the protein structures, tiny protein structures attached to the cilia. Okay, on the dendrites. Anyway, so in human, so far the number of genes those are involved in olfaction have been identified. About 10,000 genes are involved in smell. However, in many other creatures, the number of these genes could be same, but in case of human, many of these genes are in mutant or inactive form. Many of these, odorant genes are in inactive form in human, not functional. In dog, for example, most of these genes are active. That's why the ability of smell in many creatures is much more than the human, depending on how many genes are in active form and how many genes are in inactive form. Okay, dogs are 300 to 10,000 times more sensitive to human to odor. Okay, Okay, now for human, most odorant molecules are made up of the combinations of 5 kinds of atoms: carbon, hydrogen, oxygen, nitrogen, and sulfur. So these are the 5 atoms, and combination of these 5 uh, make most of the odorants in human. And that combination could be in many different, you know, ways. 2, 3, 4, or 5, and if you know how much the ratio of each type, that also even same 2 types, but the amount of each, that would change the smell. So the number of smell is not fixed. We can create new smell. So there are so many, you know, smell um, on the earth. We don't know many of those, as well as we can create new smells, and that's the beauty, that you know, when you um, you know, cook the food, you add different materials, and the even, yes, you give 10 people to cook, you know, chicken, they will cook differently, and the taste and the smell will be different, right? So that is the you know, very powerful thing in smell. You can be innovative to create new smell. Okay, so I have already mentioned the evolutionary development of the olfaction. That's the most ancient, primordial uh, sense, and we have identified about 10,000 genes. Those are involved in olfaction, and the adaptation is pretty strong in case of smell or olfaction. You know, if you go to visit your, you know, friend's house who is cooking food for you, when he opens the door, you just enter, you smell the food very strongly, right? Then after, within few minutes, it doesn't bother, or it doesn't, you know, you don't feel that strongly. It's like, you know, um, quickly get adapted. So the adaptation in olfaction is pretty fast and is strong. However, the olfaction is probably the most, you know, uh, strong perception, which is linked to the memory and emotion, and therefore the smell signal is heavily processed in those brain structures. Those are involved in uh, the memory and emotion. For example, amygdala, limbic system, right? Hippocampus. We'll talk about that. Detection of threshold of olfaction. We can measure individuals, you know, threshold, and one way to measure the threshold is you mix the odorant uh, with air, fresh air, and uh, at different, you know, amounts, and then you let the person smell the air, And what we usually do that in 1 million molecules of air, in 1 million molecules of air, we mix the odorant at different numbers, and that's why the unit is ppm, parts per million. Okay, and then we randomly, present the air to the person's nose to smell and see that which concentration the person can detect, the minimum concentration of the odorant in the air. Okay, and that is the most common way. Typically, we use this method to measure the threshold. And the person's inability to smell is termed as anosmia, a absence of smell. So normal healthy person smells something, but the person who has anosmia cannot detect that odor or smell. So that is anosmia. It can occur due to genetic disorder, lack of the genes, those are responsible for smell, so genetic, or damage of olfactory nerve fibers or pathway of olfaction. So olfactory nerve damage, or it could be anywhere in the pathway like olfactory tract or somewhere else. Okay. There is another term that is called phantosmia, phantom, ghost. So actually no existence of any source of smell, but the person will strongly smell something. So normal healthy person will not smell anything, but the person who got phantosmia will smell something. So that's why it is called phantosmia. Okay. So the odors could be many different types, modalities and submodalities. For example, fruits, we can have many different smells of fruits. So fruits could be many different smells. The cooked food, as I told you, it could be innovative to create new smell when you cook something. Even same food, but the smell would be different. How you add the odorants. So and that's why since the smell, the number of smell is unlimited, so we cannot really name them like we name the color like red, green, or blue, or yellow. We cannot do that in case of smell. That's why we say smell like banana. Many things could smell like banana, and you can create the perfume or scent that smells like banana. So artificially you can create the smell of banana. So we usually name that way, that it's orange smell or rose flower smell or banana smell, like that. Okay. As I mentioned, that the molecules, the odorant molecules must be very small and should be able to float in the air. However, it is not applicable uh, for all molecules. Sometimes, even the molecules are very small, and they have all the properties that the small molecules should have, but they might not have any smell. For example, methane is a very small molecule, methane, and carbon monoxide, similar molecule, and both are small and have the properties to be odorant molecule, but they don't have any smell. And carbon monoxide, is toxic, harmful, right? Uh, and carbon monoxide is used in uh, air conditioning or refrigeration system. So since this toxic chemical doesn't have any smell, we add the chemical that has a strong smell. So if it leaks inside your house, you can immediately tell that carbon monoxide is leaking. Okay, so we add the chemicals or molecules that have a strong smell with carbon monoxide. So in that case, you can detect the carbon monoxide. Okay, so now although human olfaction is not very strong, but still you can detect the smell, and by detecting the smell, you can detect a person, if it is male or female, by smelling the breath or the smell that comes out with sweat from the body. You can detect a female or male. So you can do that, as well as you can detect your own body smell. For example, smelling your t-shirt, you can tell this is your or someone else t-shirt is that. So we have that ability to detect the odor. Okay, so as I told you that the olfactory receptors are located inside the nasal cavity, but where? In the epithelium, olfactory epithelium. So olfactory epithelium is located here in the uh, roof or the top of the nasal cavity. Okay? And it is just below the bony structure called the cribriform plate of ethmoid bone here. Okay, so olfactory epithelium is located here, and the receptor cells are located in the olfactory epithelium. Okay. Olfactory receptors. Actually, when we say olfactory receptors, we indicate the olfactory receptor cells because the receptors are attached to the cilia of the dendrites of the receptor cells, right? And I already mentioned that the receptors are G protein coupled receptor family. And now receptors are G protein coupled receptors. Okay, and the characteristics of these receptors depends on the genes that I mentioned, and so far about 1,000, more than 1,000 genes. Probably I mentioned 10,000 in last, a few minutes ago. That is not right. Just know that more than not 10,000, 1,000 genes have been identified. Okay, and which is considered as the largest mammalian gene family. And the number of actually the genes could be same in different creatures, in human, mouse, rat, dog, roughly same size, the number. However, the number of intact or functional, or active gene is much higher in rodents and dogs compared to other creatures and human. Okay, so here you see that from the source of smell or odor, the molecules float in the air, right, and enter into the nose. Now inside the nasal cavity, in the lateral wall of the nasal cavity, you have bony elevations, and those bony elevations are covered by thick mucus membrane, and those are called concha. And those concha work as turbinates. So turbinates, the air that enters into the nasal cavity, this concha or turbinates will create air turbulence inside the nasal cavity, and the air, the odorant molecules will stick to the mucus secreted and attached to the olfactory epithelium. So in the olfactory epithelium you have a secretion that lines the olfactory epithelium, that is called the mucus, and the cilia are actually present or located inside the mucus. So the odorant molecules will first get dissolved in the mucus and then will bind with the receptors. Okay, and after the receptors are activated, transduction occurs, then the signal will go to the olfactory bulb, which is lying on the cribriform plate that I mentioned here. This bony plate is called the cribriform plate of ethmoid, and in the cribriform plate you have many tiny holes. Those are called the olfactory foramina. And this is the cribriform plate, has olfactory foramina, tiny holes, okay, for the olfactory nerve fibers. So they will pass through the cribriform plate and will enter into the olfactory bulb, which is just resting on the cribriform plate. One thing here, you just see that in case of your other special senses, the receptors are heavily protected, because the retina has the photoreceptors, and retina is in the innermost layer of the wall of the eyeball, right, and is protected by the inside the eyeball. In case of your hearing, the hair cells are located inside the cochlea, which is a bony structure, and heavily protected inside the ear. But in case of your taste, the receptors are encapsulated by the papilla. So located inside the papilla. Inside the papilla you have the taste buds, then you have the receptors inside the taste buds, so protected. However, in case of the olfactory receptors, they are not really heavily protected. Okay. And we know that most of the sensory systems, in case of most of the sensory systems, the signal from one side goes to the contralateral side, but in case of olfactory system, it stays mostly in the same side. That is another thing which is different. And also, in case of the olfactory system, the signal is not relayed in the thalamus. Thalamus is the major sensory relay station, but in case of olfaction, the signal is not relayed in the thalamus. It bypasses the thalamus. Okay. So if you just see the olfactory epithelium, so this is the epithelium here, and this is the thin layer of mucus. Okay, and you see the cilia. These are cilia, hair like soft hair like a structure, and you have the mucus, that watery secretion here from the mucosa. So the cilia are floating in the mucus, that watery secretion. Okay. Now, in the epithelium, you have three types of cells. You see these colored cells shown by 4 different colors. These are the olfactory receptor cells. Okay. also called olfactory sensory neurons. And these are the most important ones because these are the receptors. Okay, and they are neuron-like cells, as I mentioned. This is the dendrite, and this is the axon, and this is the cell body. Okay, and another type of cells, those are called the basal cells. The basal cells are actually like stem cells. They can quickly go through mitosis to produce new olfactory receptor cells or olfactory sensory neurons. Why you need basal cells there? As you know that rhinitis or infection in the nose is pretty common. It's not uncommon, and that can kill the olfactory receptor cells. So in that case, the stem cells, basal cells, will quickly produce new receptor cells. So basal cells are precursors for olfactory receptor cells or neurons. And another type of cells, these are supporting cells to support the receptor cells. Okay, so those are three types of olfactory receptor, three types of cells present in the olfactory epithelium. Okay. Now, the receptors are attached to the dendrites. So this is a receptor cell, and the receptors are attached to the dendrites, right? And the smell molecules will bind with the receptors, and that will activate the second messenger system and will create signal transduction action potential that will travel through the axon of the receptor cell. And these axons are actually olfactory nerve fibers, and you see this is the cribriform plate, and these are the olfactory foramina in the cribriform plate, right? And the axons pass through the olfactory foramina, and the axons of the olfactory receptor cells are the olfactory nerve fibers. These are very fine nerve fibers, one of the most finest fine nerve fibers in the body. Sometimes the olfactory nerve fibers can be destroyed here. If something hits your head, and if this cribriform plate displaces or displacement or movement or shaking of the cribriform plate occurs, that may destroy the nerve fibers, and in that case the person may have anosmia, inability to smell. Anyway, so you see just on the cribriform plate, this structure is called the olfactory bulb, and inside the olfactory bulb you have two types of cells, mitral cells, and another cells are called tufted cells. Here, see, mitral cells and tufted cells. Okay, and the dendrites, so these are the mitral cells. They have shown here mitral cells. You see 4 different mitral cells, and mitral cells are also neurons located inside the olfactory bulb, and these are the dendrites here of the mitral cells, and cell body here, and the axon. These are the axons of the mitral cells. Okay, so here what happens inside these round or almost round kind of round structures, these are called glomeruli. what happens in the glomeruli, singular is glomerulus. So inside the glomeruli, the axon terminals of many receptor cells make synapse with the dendrites of the mitral cells and tufted cells. So the locations where the synapses are formed, those are the glomeruli. Okay. So now you see, this is a hypothesis. You see 4 different types of receptor cells here, and from 4 different types of receptor cells, these 4 different glomeruli are getting axons. Sorry, the opposite. From each type of receptor cells, for example, this one, the cyan color or bluish, from these, only from these, the axons are going to this glomerulus, and from the purple receptor cells, the axons are going to this glomerulus. Okay, makes sense. And red going to, from all these red receptor cells, the axons are making synapse here with the mitral cell here. So and the green here. So now what that tells, that hypothesis tells that this mitral cell will process the signal that will go from these receptors only, okay, and this mitral cell will process the signal from other type of receptor cells, and many receptor cells are giving signal to a single mitral cell. So that's a convergence of the signal. Okay, so that's how different mitral cells process different types of smell. Makes sense? So this one will process one type, this one another, this one another, this one another. Okay, and this is of course a hypothesis, and however it is believed that the encoding or coding of smell is mainly occurs inside the olfactory bulb. So that differentiation of smell or discrimination, or all those are coded in the mitral cells and tufted cell. Now all the axons of the mitral and tufted cells are bundled together to form the olfactory tract. Olfactory tract. Okay, so this is the olfactory tract, and that olfactory tract takes the signal to the other brain structures. So as I told you that olfaction is heavily related not only to smell, perception or discrimination, but also related to memory and emotion. So a number of structures, brain structures, receive signal from the olfactory bulb. Okay. So this is just you can do experiment on olfactory sensory neurons or receptor cells easily, because extracting the olfactory sensory neurons or receptor cells is relatively easy, because they are in the nasal epithelium, and you can easily extract or collect them and put in a petri dish, and then in the fluid you can add different odorant molecules and see which type of odorants produce action potential. So you can record very easily. You can put the electrode on the cell body, right, inside or outside, and then add the odorant molecules and see what kind of odorant molecule stimulates this receptor cell. So that way we got all this information that there are different types of olfactory receptor cells, and since they are activated by different types of smell molecules. Makes sense? So this is the real fluorescence dye technique you can record, and also you can use electrophysiology to record the membrane potential. Okay. So I have already talked about these things. Olfaction is ipsilateral, and it bypasses the thalamus. So here you see this is a cilium, the end of cilium, and this is another. And on the cilium, on this one, you have one type of receptors, and in this cilium, you have another type of receptors, right? And these molecules will bind with this receptor, and these molecules will bind with these receptors. Okay, so that is what we believe, that on the cilia there are different types of receptors. They have different shapes, and the smell molecules also have different shapes. Right? Some are simple, some are more complex, and we have the receptors of different types on the cilia, and the binding of the signal molecules with the receptors is necessary to activate the receptor cells. And here again you see the mucus, that secretion, and the cilia in the mucus, the olfactory epithelium. In human, it has been indicated in a number of articles that human have approximately 350 different types of olfactory receptors, and each type can be activated by only a restricted set of odorant molecules. Okay. And human have 10,000 to 20,000 estimated number of each type of olfactory receptor neuron, and a total of about 3 to 7 million olfactory receptor neurons. Okay, and in the olfactory bulb, there are about 5,000 glomeruli. So you can see here the number of glomeruli is way less in number than the number of receptor cells in the epithelium, and that's why you have seen before I showed you that each glomerulus receives signal from a number of receptor cells and makes synapse with the dendrites of less number of mitral cells, or a single mitral cell receives signal from many receptor cells in the glomerulus. So the number of olfactory receptors is much higher than the other senses. For example, higher than visual or auditory. These are the results from the experiments on the mice, and on the olfactory receptors in mice, they have about 1100 different types of receptor cells, olfactory receptor neurons, and 10 to 20 million total olfactory receptor neurons, and about 1800 glomeruli, which is much higher than human. In human, we have about 500. 5,000. Sorry, in human we have 5,000. So in case of mice, the number is here looks like less than human. Okay. However, remember, these are the reports from the experiments, and definitively we cannot exactly tell the number. It has been mentioned that in dogs, the number of olfactory receptor neuron could be 1 billion, and bloodhound has as many as 4 billion. So the number, more number, indicates the lower threshold as well as the ability to discriminate, much higher ability to discriminate different odors. And the ability to discriminate the odor depends on both the genes. So genetic factor is one. How many genes are active in your olfactory system, that is one, and another is also practice or experience. So both are important. So when the receptors, G protein, coupled receptors are activated, then the ion channels in the olfactory receptor neurons cell membrane open, and calcium and sodium ions enter into the cell, and the cell membrane slightly depolarizes. So now you know that the mitral cells in the olfactory bulb are not too far from the nasal epithelium or the receptor cells. So the distance is very short. Just the cribriform plate is separating those two structures, the epithelium and the olfactory bulb. And the action potential that is generated, and the signal will move to the mitral cells or ducted cell in the olfactory bulb. And here you see that how the G proteins are activated. Okay, so this is the odorant molecule, the signal molecule. This is outside of the cell, this is inside the cell, and this is inactive G protein attached to the inner surface of the cell membrane. Okay, and this is the receptor. So the G protein is attached to the inner side of the receptors. So when the fit will be perfect here, the molecule will perfectly fit here, that will activate the G protein. Okay, and when the G protein will be activated, then what will happen here, you see that the alpha subunit of the G protein will get detached. Okay, and that detached alpha subunit will activate the adenyl cyclase. Okay, this is the adenyl cyclase, which is inactive, and when that alpha subunit will move from here, from G protein to the adenyl cyclase, that will be activated. The adenyl cyclase will be activated. Okay, and that activated adenyl cyclase will convert the ATP to cyclic AMP. Okay, and that cyclic AMP will act as a second messenger. And this second messenger is very important because this will open the ion channel. Okay, so the second messenger, which will so cyclic AMP is the second messenger, and second messenger which will bind with the sodium channels and open it, and sodium entrance or influx will occur, right? So that will cause the generation of potential. Okay, so as I mentioned that a number of brain structures receive signal from the olfactory bulb via the olfactory tract, right? So here the piriform cortex, okay, which is actually considered as the primary olfactory cortex. Okay, so this is the primary olfactory cortex, and this piriform cortex will give you the perception of smell, like primary auditory cortex or primary visual or primary gustatory cortex. So the piriform cortex will first receive the signal, and also the signal will go to the amygdala, okay, and another structure will receive the signal, that is the entorhinal cortex. So from the olfactory tract, the signal will go to these three structures, okay. So these three structures will get signal from the mitral and tufted cells. And as you know that the amygdala, this structure, main structure, is crucially involved in emotional responses, okay, and can be activated by the emotional stimuli across various sensory modalities, so including by visual, auditory, as well as olfactory, gustatory stimuli. So, you know, from all special senses, the signal will go to amygdala, and amygdala is involved in emotional responses, okay. Now what happens next? The amygdala sends signal to the hypothalamus. Okay, so amygdala sends signal to the hypothalamus, and the hypothalamus is involved in a wide range of functions to the release of hormones. You remember, probably you know that hypothalamus controls the endocrine glands. How? This is the hypothalamus and this is the pituitary, which is the master gland, right? Why it is called the master gland? Because it controls the other endocrine glands of the body. So you see, hypothalamus is the master of the master, okay? So we know that hypothalamus regulates the release of hormones, and through the hypothalamus also our thirst, hunger, and sexual behavior are also regulated, okay. So hormonal regulation as well as the regulation of those functions are controlled by the hypothalamus, and that's why it is important that hypothalamus receives some signal from the amygdala. And you know that a smell can trigger those activities and can release some hormones in the body and arouse some you know, functions or desire. And then the entorhinal cortex, this one is the gateway. It is called entorhinal cortex is the gateway to the hippocampus. So from here signal mostly goes to the hippocampus, which is widely known as the site for memory, long-term memory storage. Long-term memory, okay, storage. Okay, so you see that from the olfactory bulb through the olfactory tract, the signal goes to the piriform cortex, which gives you the immediate sensation and then perception. Sensation is like, you smell something. Okay, when you enter into your friend's house, you smell. Immediately you can tell that something, you smell. So that is the sensation. Then you start to, immediately after that, you realize that is it from pizza or chicken or, steak or whatever. So that is the perception. Okay, so those are given by the piriform cortex. So this is considered as primary olfactory cortex. Then also some signal goes to the amygdala, which is heavily related to emotion, and from there goes to the hypothalamus. And then also signal goes to entorhinal cortex, which the shape is like the elephant nose, rhinal nose, and that structure, the entorhinal cortex, that one is the gateway of hippocampus, which is very known for long-term memory storage. So you can see that if you smell something that you used to eat that food, many years ago when your mom used to cook that food for you, you not only smell that, it quickly triggered the emotion as well as memory. Those okay. Every time I think about movie theater, I smell like popcorn. So it's pretty strongly related to the memory and emotion system. Okay, and so we talked about these structures already. I'll add a few pages. I'll send separately to give you better idea how the function is processed in the brain, the olfaction. Okay. This one is a better cartoon picture that shows you different brain structures better. So you see the location of amygdala, okay, entorhinal cortex here. This is the piriform cortex, the yellow one, pretty big, okay, and this is hypothalamus here. This is hypothalamus. Thalamus is here, so under the thalamus, hypothalamus, right? And hippocampus, this memory storage site, is here. Okay, so those are a few very important brain structures that are activated or they are responsible for processing the smell. And one more structure that is called this structure here, you see that green. This is the orbitofrontal cortex. This is also involved in the processing of smell signal. Okay, so just know those structures, and now here you see this is the olfactory receptor neurons, We have the olfactory nerve fiber. Signal goes to the olfactory ball, where you have the glomeruli, the synapses, and mitral cells, as well as the tufted cells, receive the signal, and then through the olfactory tract, the signal is taken to the piriform cortex, which is the primary olfactory cortex, as well as amygdala and entorhinal cortex. Okay, and from amygdala, signal goes to hypothalamus. From entorhinal cortex goes to hippocampus. Okay, orbitofrontal cortex receives signal from all these three structures. So from all these three, the signal goes to, here you see the orbitofrontal cortex here. Okay, so that's the pathway of smell signal or olfactory signal. Now we know that olfactory nerve is the cranial nerve number 1. Cranial nerve number 1, and it is carrying the smell signal to the brain, right, from the nasal epithelium or receptor cells. Another nerve which is also involved in smell, particularly the smell that gives you the burning sensation, like gasoline or hot pepper. Hot pepper or gasoline, it's a burning sensation, but that also gives you some kind of smell. So when you eat those hot pepper, or you smell the gasoline that burns your sensory neurons, or activate your sensory neurons, that would be more appropriate, not burns, activates the sensory neurons. That signal from the sensory neurons also contribute to your smell, and that signal from the tongue or from other areas go through the trigeminal nerve. So that is somewhat a sensation, right? Like burning sensation in the skin. So trigeminal nerve is the cranial nerve number 5. Okay, so those are two nerves. There are two theories how the olfactory receptors are activated. One is called shape pattern theory, which is more widely accepted theory. And I talked about that before, like the odorants have different shapes, right? Different odorants have different shapes, and different receptors on the cilia have different shapes, binding sites of different shaped binding sites. So now here you see permutation combination. There could be many different types of odorants. Even the shape, all these three have triangular, same shape. All these three have rectangular, right? However, they could be different. Okay, so not only the shape that determines, but there could be other genetic, factors, whatever. So you see, there are 9 different odorant molecules and 3 different shapes of them. Okay, and here the receptors could also be different. Binding sites are same, like same shaped binding sites, but they are not exactly same. Okay, so what happens here, this hypothesis says that the binding should be exact exact means in terms of shape as well as in terms of the nature, that means the color. Okay, so here most of the odorant molecules are compounds, that means the combination of different molecules. Okay, so this is a compound, odor compound. It has two different molecules, right? This one has two, this one has three, this one has three. Okay. Now these are the receptors here. So we'll see which receptor here will be the best for this odorant compound, or will be activated most, or will be most suitable for this odorant compound or molecule. So here you see by if you see here this triangular will bind with this one as well as with this one. So this triangular part, the bottom part, will bind perfectly with these two, right? But that will activate these two differently. Why? Because you see here the color blue and this one has blue color, right? So both matched, the shape as well as color. Shape and color. That is the nature or type. And here another receptor will be activated by this one the best. You see when this rectangular portion will bind with this and these two receptors, this one will be activated more than this one. Why? Because the color, this rectangular molecule is yellow and this receptor has the shape rectangular as well as the color, so both will match here, the color and shape. So here this one we see will activate this one the most because yellow as well as the shape of this. Makes sense? So that's how best activation is shown by the green color here. Okay, some activation by yellow and no activation by red. So here you see no activation by red because the shape doesn't match at all. So no activation. First shape should be perfect fit, then also the color. So that is the shape pattern theory. Now vibration theory is less well accepted by the researchers or scientists. What it tells that every small molecule, all molecules have vibration. Molecules vibrate at different frequencies, and that determines the activation of the receptors. So the frequency of vibrational frequency. Different molecules have different vibrational frequencies, and that would activate different types of neurons. Okay, this theory is less popular than the shape pattern theory. I talked about this before, that you can create a completely different color by combining two or more colors. For example, long length color red and medium length color green. When you combine them, will give you yellow or orange, and that is completely a new color. There is no information if you don't know. If I show you yellow, you will see, you'll tell this is yellow. You don't see any other thing there. It's completely yellow. If you know before that these two colors create yellow, that is a different thing. But if you see yellow, you cannot separate the green and red in it. Okay. So synthesis, completely new color is produced here. You cannot discriminate from discriminate the two or more colors in it. Olfactory is not purely synthesis, it is a mixture of analysis and synthesis. In analysis, you can separate. If you are careful, you spend time and focus, you can separate different components. In synthesis, you cannot separate like this one. Example of analysis is auditory. When you hear the music, if you are careful and hear carefully, you can separate different instruments there. Okay, that which sound is coming from which instrument. You can do it in some extent, but in case of sight or vision, you cannot do that. So this is synthesis, and in case of auditory, you can do analysis. Discriminate. You can do analysis. In case of vision, no analysis, only synthesis. However, in case of olfaction, it is actually a mixture of synthesis and analysis. So you can actually discriminate in some extent, not as good as auditory, but you can discriminate that combination of different smells are in this smell, although it is a different smell. The term pheromones. This is a strong, stimulant that can trigger the discrete behavior. So in the late 1950s, the term pheromone from the Greek word pherein, which means to transfer, and hormone, that means to excite. So that is pheromone that can strongly trigger the behavioral response. Okay, and for example, female silkworm moth emit a chemical called the bombicle, and this bombicle can elicit a strong behavior in the male moth. Okay, and that attracts the male. Also, elephants secrete a pheromone that can strongly attract the male and female to each other. Okay, so pheromones used by many animals to find the mates. So it is said that only 200 molecules of pheromones are sufficient to elicit a change in behavior, and that is a strong, signal. And it is also said that one molecule is sufficient to elicit a neural response. So to activate the neuron, one molecule is can do that. Okay, so it's a very small, just one molecule of pheromone can trigger the action potential in the neuron. Although you won't realize anything, you won't feel anything, but the neuron will be activated. However, to give you the sensation and perception and trigger the response in your body, many neurons should be activated, right? So, pheromone. Just read this, so you'll understand. So this is experimental result that population code for odors, and 11 different mouse, the response of the olfactory receptor neurons from 11 different mice were uh, recorded, and the response of neuron was uh, very different. Even they used 4 different types of or groups of chemicals. They have uh, different carbon chain length. The length 6 carbon chain, 7 carbon chain, 8 and 9. Okay, so these are 4 different groups of chemicals, and in each group you have 2 different chemicals, and both have 6 carbon chain structure. And you can clearly see the response here. These are the neurons, 1, 2, 3, all the way to 11 neurons response. So no response, no response, no response. And neuron, only one neuron, that is mice, in mouse 4, the neuron responded to this chemical. Okay, hexanoic acid activated the neuron in one mouse, and hexanol, another chemical that has same configuration, 6 carbon chain, activated totally different mice. So you see that these two different response. These two, although they are structurally very similar, activated different neurons of different mice. Here you see adaptation experiment. So if you record the evoked potential by activating with the odorant in short interstimulus interval here. You stimulated by brief pulse, which lasted 0.01 second, by the same odorant. So you presented in short interval, and duration is 0.01 second. And if you do that in a brief interval, like 1 second interval, then you see the second response is much weaker. That means the adaptation is still there. Now if you increase the interstimulus interval to 5 seconds, then what happens, the receptors get free again to bind with the molecules again, and you see the strong response. So here you see adaptation. Here in long interval we don't see adaptation. In real world you experience this if you stay inside the house, and you are cooking something, so your olfactory system gets adapted to that, so you don't smell that. Now if you quickly get it out and come back, still you won't strongly smell that. But if you go to fresh air and spend a little time, then come back, then you will smell more strongly. Okay, so those are the experiments. You can see that you can study the smell perception and the response of the neurons and activation of the receptors. All those kinds of experiment can do. Here you see the response of mitral and tufted cell in olfactory bulb. and recording was performed by optical imaging. And the pattern of activation of olfactory bulb on the upper panel, optical imaging responses to 4 different odorants with similar molecular structure. Now this is something, this result is opposite of one of the previous one that I showed. Here you see that different odorants, but they have similar molecular structures. Activation in the olfactory bulb neurons. However, remember these are olfactory bulb neurons, mitral or tufted, not in the olfactory receptor cells. So here you see that the same neurons are being activated by similar type of stimulus. And lower panel responses to some odorant at different concentrations. So this is the intensity activation. More activation, higher concentration. Higher concentration, more activation. That makes sense. The density or concentration of the odorant will trigger different responses if you change the concentration. So this is higher concentration at 10%. This is 2%, 1%. This is 0.1%. So you see the activation is much stronger here. So that is the intensity of the response. The adverse odor or bad smell can trigger like sulfide cocktail, can trigger the amygdala. This is recording from responses in the amygdala, okay, and orbitofrontal cortex. I showed these 2 areas before. So you see that this is amygdala. So that disliking or strong adverse or disliked smell can strongly activate the amygdala as well as orbitofrontal cortex. Here you see the adaptation again. This is the sensitivity of the olfactory receptors. The sensitivity of the animal, this mice, let's see, rats, not mice, so rats, experiments on rats, and the sensitivity was determined by the investigation time. Like this is the case, and this is the animal here, okay, the rat is here, and here you put the odorant, okay. So if you put the odorant, the rat will go there and will try to find what smell is that, okay. So how much time it is spent to find that smell has been plotted here by second. So here you see first time when you present the odorant, the rat spent a lot of time, about 10 seconds to investigate or identify that. Then in second trial, if you present the same odorant, that is called adapted odorant again, then it will spend less time. Then gradually you see that it spends less and less time. So it got the information, and it doesn't need to spend time as long as it was needed at the beginning, the first trial. So you see here at 5th trial, the time it is spent there is about 3 seconds. Okay, so that is another behavioral adaptation, we can say, not the neuronal adaptation. Okay, now if you give a new type of order there, then it will again spend a lot of time. That is the test order. Okay. Another term that is called cross adaptation test. The rat is first adapted to acetic acid. That is the adapted odorant. Okay, acetic acid here, and you see the investigation time here. And then rat's investigation time is measured for 5 different taste odorants, including the acetic acid. And the taste odorant were different only in their carbon chain length. Okay, and the investigation time increased as the carbon chain length increased. So this is propionic acid has longer carbon chain than this one, acetic acid. This one has longer, this one has further longer, and caproic acid, this one has long, very long. So longer means more complex, right? So that the animal spent more time to identify that and get more information. And as the molecule is more complex, that animal will spend more time. So that's how the behavioral adaptation tests are performed. Just one example. Okay, so those are the things you need to know from the smell or olfaction. In next lecture, I'll talk about the taste system, which is the gustation.